All right, let's go ahead and get started. So the only announcement I have, I'm sure you're all aware, next Tuesday is a school holiday, so there will be no class next Tuesday. I know you're so sad to miss class, but yeah, so we'll have class this Thursday and then not Tuesday, and then we'll get back to normal schedule. All right, so let's review what question, yes? Yes, I think Monday is not a holiday, right? Yeah, so yeah, sections will still happen this Friday, will still happen this Monday. There will be new homework posted this Thursday, so it would be back to the usual thing. Yeah. Other questions? All right, so let's review what we talked about in class last Thursday. So we talked about what has to happen to that initial transcript that is made by RNA polymerase II in the case of a pre-messenger RNA. So the pre-messenger RNA is going to be that nascent or that original transcript that got made by RNA polymerase II. And as we saw, there's multiple processing events that need to happen to mature that messenger RNA. All messenger RNAs get capping on their 5' end. There's that M7G, that guanine that is methylated on the 7 position, that gets attached through that triphosphate bridge to the 5' end. And that serves 3 very important purposes for the messenger RNA, which are, what is one? Stabilize, translation, and export it out to the cytoplasm, exactly. Most eukaryotic messenger RNAs also undergo processing at their 3' ends. There are polyadenylation signals that tell the machinery where to cleave the messenger RNA, marking the end of the 3' UTR, and then add that string of adenosines. And in humans, it's usually about 200, 250 A's that are added to the 3' end in that polyadenylation event. And then we saw once you have that poly A tail, there are proteins that bind to that poly A tail, and they are called poly A binding proteins. And the poly A tail also serves 3 very important purposes. It helps protect that 3' end, helps get that messenger RNA out to the cytoplasm, and then we'll learn later today how that poly A tail bound by poly A binding protein actually helps in the initiation of translation. And finally, we talked about splicing. So splicing is simply the act of removing the introns and ligating together the exons. And we saw that the complex that carries out splicing is called the spliceosome. It consists of over 100 proteins, as well as small nuclear RNAs, or snRNAs. And we saw that 2 of those snRNAs play an important role in helping to identify splice sites. We saw that U1 snRNA base pairs to which splice site? So there were 4 splice sites we talked about. U1 snRNA base pairs to, take a guess. 5' splice site, exactly. So U1 snRNA base pairs to the 5' splice site. And U2 snRNA then base pairs to the branch point A, exactly, and helps position for that first transesterification reaction to occur. We also talked about that there can be alternative splicing, and what we mean by alternative splicing is different exons can be chosen to be included in the final mature messenger RNA. And what that means is a single gene that produces a single pre-messenger RNA can actually end up making multiple different types of mature messenger RNAs that can then encode for multiple different types of proteins. So it really expands what our genome is able to encode for. And finally, once you have that mature messenger RNA, it can be it can be exported out to the cytoplasm through those nuclear pore complexes, those big protein complexes that are inside the nuclear membrane. The messenger RNA goes out to the cytoplasm, and there it can be translated into protein. And we'll talk about translation later in class today. Any questions about messenger RNA processing? All right, clicker question for you. Above is the gene Y pre-messenger RNA, and next to it is a northern blot for gene Y RNA in 4 different cell types using a probe complementary to the end of the 3' UTR, so just where it's showing right here. Note the mature mRNA gets a 200 nucleotide poly A tail, and so the results support which of the following? And 10 more seconds. Get your final answers in. Ooh. All right, so we have a little bit of everything. Not a lot of love for D, though. Interesting. All right, let's go through each one of these. Oops, that's not what I wanted to do. All right, who wants to share their answer and thinking? Khalil? Is it? Yes.
I chose D because apparently N is correct.
Okay, so let's go through these. So Khalil likes more than one of the above. That's often a good answer, not always a good answer. So you liked A because A said alternative splicing skips exon 3 in the D cells. And why did you like that answer?
It was like in the northern blot there is nothing, so I assumed it was skipped.
So I like your reasoning in that you don't see anything, and we see exon 3 is where our probe binds. But can terminal exons be alternatively spliced? So alternative splicing means instead of this 5' splice site ligating to this 3' splice site, it would skip that and ligate to this 3' splice site. Can terminal exons be alternatively spliced out? No, they cannot. You can have alternative transcriptional start sites. You can have alternative polyadenylation sites. But there's no way you can skip by splicing this last exon or the first exon. Does that make sense? We understand alternative splicing? Yes? So if we had the 5' splice site here, it needs to ligate onto a 3' splice site. If it skipped exon 3, there is nothing for it to ligate onto, so that would not be a splicing event. So you would need a 5' splice site, can skip that 3' splice site, and then go to this 3' splice site, skipping exon 2. So you can skip internal exons, but not the terminal exons, because you would be missing either a 5' or a 3' splice site. Does that make sense now? It's a little bit tricky, but as long as it makes sense now, right? All right, so A is not correct. What about B? SR proteins that recognize ESCs in exon 2 are absent from the C cells. What are ESCs? Use that term in the last class. Yes? An exon splice enhancer. And what do they do? What's an exon splice enhancer do? Yeah, exactly. So these RNA binding proteins, called generally SR proteins because they have a lot of serines and arginines in them, they bind to these exon splice enhancers inside the exon and sort of decorate the exon and say, hey, I am the exon here, and so the splice sites next to me are the correct ones to use. So when you bind to an ESC, you are promoting inclusion of that exon in the splicing event. All right, so this says SR proteins that recognize the ESCs in exon 2 are absent from the C cells. So if they were absent, what would happen to exon 2? It would get skipped, right? So if they were absent, you would skip exon 2. All right, if we skipped exon 2, what would be the size of our mature messenger RNA? 550? If we skip exon 2? 450, right? So now we look in our C cells. Ooh, do we have a 450 band? No. So that is not consistent with what we are seeing for the C cells. Does that make sense? Everyone remember what an ESC is and what SR proteins do to keep the exon included? Question? So you have 600 right here, right? If you get the polyadenylation with the pre-messenger RNA, and then you subtract 50, 50, 50, so you subtract 150 from 600, 450, right? Does that make sense? So B is not correct. What about C? Exon 2 is skipped during splicing of gene Y pre-mRNA in the A and B cells. Well, we just said what would happen if exon 2 is skipped. We would get what size? 450. Is that what we see in both the A and B cells? No. If I just said A cells, would that be correct? Yeah. And do you understand why we can have two bands here? You can have two different splicing events in the same cell. Remember, lots of that pre-messenger RNA is being made, and maybe those cells have half the amount of SR proteins that are needed to recognize those ESCs. So half the time it's recognized and you get the right size RNA. The other half of the time it's skipped and you get a different isoform. Does that make sense? All right, so C is not correct. E was not correct because it's not more one than the above. So it is D. Gene Y undergoes alternative polyadenylation, producing a 50 nucleotide 3' UTR in the D cells. What do we mean by alternative polyadenylation? Yes? Yeah, you use a different polyadenylation signal in the 3' UTR. And so instead of using this position to cleave and polyadenylate, if you use another polyadenylation signal that's only 50 nucleotides away, you would get cleavage there, right? Because the whole 3' UTR is only 100 here. If you cleave there and this was your remaining messenger RNA, would your probe see that RNA? No, because that part that the probe binds to would have gotten chopped off. It would no longer be part of this alternatively polyadenylated messenger RNA. We go to D, we see no messenger RNA in the D cells, and that's exactly what would happen if we cleaved and polyadenylated and didn't include the entire 3' UTR, which is where our probe needs to base pair to. Question?
So are there 200 adenosines after the probe, or is
If you cleaved here at 350, what would then happen after the cleavage event? Whenever you cleave, then what do? You polyadenylate, right? So if there was a weak, let's say a weak polyadenylation signal that's here, that usually gets skipped and so goes all the way out here, you cleave here. After the cleavage event, that's when poly A polymerase hops onto that 3' end and adds the poly A tail. In these cells, we got cleavage at this position. Poly A polymerase will jump on at that position, add a 200 nucleotide size tail. Does that make sense? Yes. Question? I wanted you to recognize that if it was 50, it would be about at this position, and so it would be far away from where the probe would base pair to. Does that make sense now? So alternative polyadenylation simply means you're choosing a different position to cleave that messenger RNA and then add the poly A tail to. Any questions about this? And so here's all the math. If you wanted to figure out all the various different sizes, here's what we just talked about. If we had alternative polyadenylation, so to 350 position, because I told you a 50 nucleotide 3' UTR, you would then add the poly A tail, 550 before splicing, 450 after splicing, but you can't see it because your probe is at that position. If I move the probe, you would be able to see it, right? Any other questions about that? All right, so before we can talk about translation, we need to talk about how we make the other RNAs that are necessary for translation. So you remember we mentioned that in eukaryotes, there are 3 different RNA polymerase complexes. Before the midterm, we only talked about RNA polymerase II, which makes our messenger RNAs. Tonight, we'll move on and talk about RNA polymerase I as well as RNA polymerase III. So here's back to our table. So RNA polymerase II is responsible for making messenger RNAs as well as snRNAs and siRNAs and microRNAs, which we'll talk about next week. RNA polymerase I makes one RNA, one long pre-ribosomal RNA that then gets cleaved into 3 separate ribosomal RNAs that are parts of the ribosome, and they are essential for translation. RNA polymerase III makes tRNAs, which we need for translation, as well as one more ribosomal RNA, this 5S ribosomal RNA, which is also a ribosome component. Also makes the U6 snRNA and some other RNAs we're not going to talk about. All right, so RNA polymerase I transcription, as I mentioned, it makes one type of RNA, and that is the pre-ribosomal RNA. There are multiple copies of this pre-ribosomal RNA throughout our genome, usually over 100, because remember, we need to make a lot of these ribosomal RNA. So it's one of these tandemly duplicated genes because we need lots of ribosomes in order to do protein synthesis. The gene for this pre-ribosomal RNAs has some specific upstream elements. It has core elements that are going to be recognized by RNA polymerase I, along with general transcription factors. One of these transcription factor components, SL1, actually includes a protein we've heard about before, TBP. Remember, TBP is TATA binding protein, needed for RNA polymerase II to recognize core promoter elements and transcribe. Turns out TBP is also needed for RNA polymerase I. RNA polymerase III. So we said that this polymerase complex is responsible for making our tRNAs, our 5S ribosomal RNA, as well as a U6 snRNA. Some of these genes include sequences that we consider to be internal promoter elements. What we mean by internal is it's part of the RNA that actually gets transcribed itself. So if transcription starts here, these A and B are core promoter elements, but they will actually get transcribed as part of the tRNA. That's what we mean by internal. And just like all of our other RNA polymerases, there are going to be specific general transcription factors that help position RNA polymerase III. We call these generally TF3s for RNA polymerase III. And one of these, actually TF3B, has TBP as a constituent. So TBP is required for transcription by any of our RNA polymerases. It's a part of the complex for any of our 3 polymerases. All right, so once we make these RNAs, there's actually quite a bit of processing that needs to happen in order to mature them so that they can function in translation. The synthesis and processing of the ribosomal RNAs happens in a subcompartment in the nucleus called the nucleolus. This long pre-ribosomal RNA we'll see needs to get cleaved into 3 separate ribosomal RNAs. Also, they get some modifications, and then we'll talk about tRNA maturation. So here, this big blue, this is the nucleus, and within the nucleus, there will be a subcompartment known as the nucleolus. It's not membrane bound, it's not really separate from the nucleus, but it's really a concentrated center where RNA polymerase I will transcribe that pre-ribosomal long rRNA, and that's where the RNA processing machinery will be to chop that long pre-ribosomal RNA into each of the mature 18S, 28S, and 5.8S. And actually, ribosomal proteins that, of course, get made in the cytoplasm, many of them get imported into the nucleus and then associate with the ribosomal RNAs and then are exported as the separate 40S and 60S ribosomal subunits. So the initial pre-ribosomal transcript, which is made by which polymerase? Other one. 1. So RNA polymerase 1, you can easily remember it. RNA polymerase 1 makes one type of RNA, this long pre-ribosomal RNA. It's about 13,000 nucleotides long when it's first made. And through a series of processing steps, eventually you get the mature 18S, which is about 1,900 nucleotides, as 5.8S, which is 150 nucleotides, and 28S, which is 5,000 nucleotides. And then these yellow parts, these spacer parts, get degraded. S just stands for sedimentation. Don't try to add up these numbers and make them add up to 45S. It's based on a physical property, not in size or addition type of size. In addition to the pre-ribosomal RNAs getting processed into their mature sizes, they also undergo quite a number of modifications, so changes to the bases. Excuse me. The most common modifications that happen to these ribosomal RNAs are shown here. What is this type of modification called? Yes, excuse me, methylation. It is on the 2' position here, right? So it's 2' O-methylation. Can it also happen at the 3' position in this RNA? What's going on at the 3' position in RNA? Phosphodiester bond, right? So this 3' is not a hydroxyl, it is part of a phosphodiester bond in a chain of RNA. 2' hydroxyl, that's available. That can undergo this methylation. What about this sort of weird-looking nucleotide here, right? So you hopefully remember that normally it's the nitrogen that is attached to the sugar ribose, and here it's sort of like twisted a little bit. Does anyone know which base this kind of looks like if it wasn't twisted like that? It's U, yeah, uridine. Because it's twisted, it's then called pseudouridine. So if you twist this back and put the N at this position, you would have uridine, sort of like uridine, so it's called pseudouridine. And this was the 2' O methylated version. So how are these cleavage events and as well as these modification sites recognized? So these modifications are very specific for a very specific nucleotide in this thousands of nucleotide long RNA. Turns out that there are small nucleolar RNAs that guide enzymes to those specific positions to cause the modification to happen. Remember when we talked about splicing of pre-messenger RNAs? We talked about how the U1 snRNA guides the spliceosome to the 5' splice site by base pairing to it. Similarly, these snRNAs, so shown in blue here, will base pair to positions, specific positions in the pre-ribosomal RNAs, and then recruit specific modifying enzymes to either cause methylation or pseudouridylation at that particular position. And so these snRNAs are base pairing to specific sites, giving the specificity. Why do you think we call them small nucleolar RNAs? Where does ribosomal RNA transcription, processing, modification happen? Nucleolus. That's where you're going to find these types of RNAs. They're going to be in the nucleolus to do these modifications to the ribosomal RNAs. All right, what about tRNAs? So they also go through a number of maturation events. So the initial pre-tRNA will undergo trimming at its 5' end. There's an activity called RNase P that will chop off this, what we call the leader sequence. The 3' end also undergoes trimming, and there's actually an enzyme called the CCA Adding Enzyme. It's a good name for it because what it does is then it adds specifically CCA onto the 3' end of every tRNA. And this is important because, as we'll see later in class, it is at this 3' end where the amino acid is going to get attached so that that tRNA can be used in translation. Some tRNAs also have a sequence, an intervening sequence, that needs to get removed or spliced out, meaning this blue sequence would be removed and then these nucleotides would be ligated back together. Looking at the sequence here, do you think that the same spliceosome that removes introns from pre-messenger RNAs is going to be involved in splicing the tRNA? See a 5' splice site, a branch point A, a polypyrimidine tract, a 3' splice site, all in this little sequence here? No. It's a completely different enzyme that works on the tRNAs as opposed to. So that's why I put splicing here in quotes. We still call this splicing because it's removing this intron and ligating the ends back together, but it is not done by the spliceosome that does splicing in our pre-messenger RNAs. And then the final thing that has to happen to a tRNA is there's a huge number of base modifications that happen. For example, there's pseudouridine that happens at several positions. There's methylation on several groups. And all of these trimming and modifications are very important for the tRNA to fold into the correct structure so that it can be recognized by the enzymes that need to put the amino acid on the 3' end, and so that it can fit into the ribosome to allow translation to occur. All right, so we know a lot about the enzymes that do all of these different events, but how was this actually determined? So how would you find the factors that carry out a particular processing event on an RNA? In other words, if you have this as your pre-RNA and you know that this gray spacer needs to be removed and these two ends ligated back together, how do we find that activity? Well, in general, what you can do is you get your candidate proteins, or maybe you want to just test nuclear extract versus cytoplasmic extract first. And so you have what you want to test. And then you make the substrate. So in this case, it would be this RNA. You can make this RNA in vitro that has a spacer sequence. In one tube, you'll add protein A. The other tube, you add protein B. And then you just need to analyze, did any activity happen? Did the reaction that I'm expecting happen or not? And so from these results, which protein had the activity? A, exactly. And so this is the general setup for how you would ask this question. And it was really asking a very simple question like this that led to a very surprising finding. So Tom Cech's lab knew that there was this type of pre-ribosomal RNA found in tetrahymena that did need to remove this spacer and ligate these two ends together to make the mature ribosomal RNA. And the reason why they were studying this reaction using tetrahymena is it turns out that this ciliate has 40,000 copies of this pre-ribosomal RNA. So whatever the activity is that is doing this reaction, there must be a lot of it in this cell type, right? In fact, we talked about tetrahymena before, earlier in class. Does anyone remember what was discovered also in tetrahymena? Telomerase, exactly. And it was the same idea. Remember that this tetrahymena has thousands and thousands of linear chromosome ends. So to maintain all those telomeres, there must have been a lot of the activity that does that. And that's where telomerase was first discovered by studying it in this tetrahymena. All right, so how did the Cech lab set up this experiment? Well, so they figured tetrahymena must have a lot of this activity, so they made an extract from their tetrahymena. They made the test pre-ribosomal RNA in vitro. You can synthesize RNA in vitro. And then they knew that if the reaction happened, if the spacer was removed, and the spacer is 600 nucleotides long, and these two ends get ligated together, you should go from 1,000 nucleotide RNA to a 600 plus 400, exactly. If these get ligated together, you'd have a 600 and 400. And you can separate those RNAs based on size. So you can just run them in a denaturing gel. You can make this ribosome or this RNA radioactive and just look, did the reaction happen? And that's what they did. They ran some controls and the results on a gel and got a very surprising result. And so let's do a clicker question and see if you can make the same conclusion that they had to come to here. So here is the input RNA. Here is, let me move that little cloud. Here's what was added in each tube. Here's a marker showing you sizes. And so what are you going to conclude? Ten more seconds. Final answers. All right, a lot of people like answer D. Who wants to share their reasoning? Sure.
I chose D because...
Which one did you choose?
D.
D?
Yeah, because as we can see in the top where it says pre-rRNA and protein extract, it seems that all the bands are identical, so it seems like it's not required that protein extract.
Absolutely, and this was a shocking result for the lab, right? Because proteins are enzymes. You need proteins to do catalysis. But here what they were finding is when they just put the RNA itself in the tube, it went from the substrate size, 1,000, to the predicted product sizes, without the need for adding any protein to the tube. This is one of the first examples of what we now call catalytic RNA. The RNA itself can carry out a reaction. And the poor grad student who kept getting this result in Tom's lab had to do so many experiments to prove to him that there was no protein contaminating her tube, because this had been unheard of before. But we now know that catalytic activity by RNA actually is much more common than we would have expected. So does everyone understand why D is the answer? Any questions about the other options? All the other options suggest that the protein is doing something. Clear on that? Yes?
So like RNAs, basically, they can do it themselves?
So RNA can fold into a structure that can promote catalysis. So remember, RNAs can base pair to themselves. They can form shapes that will then induce catalysis. And so, yeah, this is the key lane here. No protein was added. It was just the RNA itself, and you got the products. And so this was, as I mentioned, one of the first examples of catalytic RNA. And this was, I met Tom Check at a meeting a couple years ago. The spacer sequence from tetrahymena being removed without the aid of proteins. And around the same time the Czech lab discovered that, workers in Sid Altman lab discovered that this complex called RNase P, we just mentioned RNase P a few slides ago. What does RNase P do? Anyone remember? Taking off the 5' end, or the 5' leader from the tRNA. So RNase P was known to be a complex that consisted of a protein and what was called M1 RNA. And it was always assumed the protein is doing the catalysis. But what they finally figured out is actually the protein is just stabilizing the RNA. The RNA is actually inducing cleavage of that 5' leader of the tRNA. And so these were the first two examples of catalytic RNA. We now know that RNA has a lot of tricks and can fold into structures to cause catalysis. In fact, peptide bond formation that occurs on the ribosome linking the amino acids together is actually catalyzed by the ribosomal RNA that's part of the ribosome, not those proteins that are part of it. And we know many other examples in our cells now. And by studying these RNAs and understanding how they can fold to induce catalysis, people are actually able to design what we call ribozymes, so ribo for RNA, zyme for an enzyme. And they have been able to, for example, design them so that they can fold into a structure and cause catalysis, and actually cause catalysis of an RNA. For example, it can be designed to base pair against. So maybe this is a viral RNA you want to cleave, you make a ribozyme, it will go in and cause cleavage of that RNA, and that's what we mean by a ribozyme. All right, so now we know all of the RNAs that are needed for translation, and so we will talk then about this last molecular mechanism, the process of decoding RNA to synthesize protein. And so first we will review what we mean by codons, how the messenger RNA is read, how the reading frame is set. When do you start reading in those groups of threes or those codons? We know that there are generally 20 amino acids, and therefore we need 20 what are called Aminoacyl-tRNA Synthetases. These are the enzymes that are going to attach the correct amino acid onto the correct tRNAs. And then we will define what we mean by wobble positions for the codon-anticodon interactions. So translation really requires three types of RNA. First, you're going to need your messenger RNA. This is the code, and it is read in groups of three. Those are called the codons. The messenger RNA has the codons. They are then read by the tRNA through a specific position called the anticodon, because it is going to base pair to the codon. That's how the tRNAs are able to read the sequence in the messenger RNA. And then finally, the third type of RNA that's required for translation are these ribosomal RNAs that are structural components and, in fact, also an enzymatic component of the ribosome. All right, so what we mean by codons, this is by reading those nucleotides in groups of 3. And so this start codon is almost always going to be AUG, which happens to encode for the amino acid methionine. The next group of 3, in this case, and we'll look at the genetic code in a few slides to see how UGU encodes for cysteine. Then you'd read the next group of 3, next group of 3, next group of 3, until you hit what's called a stop codon. There are 3 different sequences that are not read by a tRNA. They do not encode an amino acid. Instead, they recruit release factors to terminate translation. If I give you a sequence and ask you to decode it to tell me what the amino acid sequence of the protein would be, we'll look at the genetic code in a moment to see how to do that. But when you get to the stop codon, don't write stop at the end of your peptide. Stop is not an amino acid. Just stop writing. This is your last or C-terminal amino acid for this particular example here. For most messenger RNAs, the first 3 nucleotides are not going to be AUG. You're not going to immediately start translating. As you'll see, the ribosome actually will start scanning from the 5' end until it finds the first AUG, and only then is the open reading frame set to be read in the next groups of 3 until the end. So all this sequence that is before this start codon, what do we call that sequence? The part before translation has started. The 5' UTR, the 5' untranslated region, because you don't start translation until you find that first start codon. What about downstream of the stop codon? What do we call this sequence? The 3' UTR, the 3' untranslated region. All right, let's make sure we all understand how to read these codons and know which amino acid it is coding for. So this is the pretty much universal genetic code. What we mean by universal is AUG is going to encode methionine if you are in a human cell, if you are in a Drosophila cell, if you are in a bacteria cell, if you're an amoeba cell. The code is pretty much universal. All right, how do we read this genetic code? So starting from the 5' end, if at the first position you have an A, then it's going to be one of these choices. So then you look at the second position. If the second position is a U, we've narrowed it down further. And if the third position is a G, we then have A, U, G. It's going to be methionine. So you're starting from the 5' end, reading the first position, then the second, then the third. Here are our 3 stop codons. So if you have at the first position U, then an A, then an A, that's the stop codon. A U, an A, and a G, that's a stop codon. U, G, and A, that's a stop codon. Does anyone have any questions about how to read this? On the exam, you will be given this code. Do not memorize it. It will be there for you to just make sure you know how to read it. And then for fun, you can go back to the last slide and try to decode that one. All right. We also sometimes refer to the code as being degenerate. And the reason why it has to be degenerate is if you do the math for 4 different amino acids, you need to have 4 different nucleotides. You need to have 64 codons. Turns out that 61 of them encode for amino acids. What are those other 3 encode for? The stop codons, exactly. But there's only 20 amino acids. So what that means then is more than one codon can specify the same amino acid. So an example here is for serine. So UCU encodes for serine. So does UCG encode for serine. So does AGU encode for serine. So there are multiple different codons that all specify serine. It's degenerate because there's more than one codon for the same amino acid, but it doesn't work the other way, that there are not multiple amino acids encoded by the same codon. So that's just what we mean by degenerate, is there is more than one codon for the same amino acid. All right, so now let's talk about reading frames. So as we said, the way the reading frame is going to be set is by scanning until you find that first AUG. And then you simply read in groups of 3 using that genetic code to know what amino acid is encoded for. What would happen if we had a mutation that removed this G? Well, that G is no longer there, and so we're going to shift frame. This would then be called the moving into frame 2. And look what happens. You now encode for an entirely different protein. By just removing one nucleotide, we have changed how these groups of 3s are read. If we lost a second nucleotide, we'd then be shifted to the next frame, frame 3. That would then encode another different set of amino acids. What about if we lost 3 nucleotides and shifted to frame 3? What would happen? Exactly. You would just lose those 3, so you would lose one codon, but then you wouldn't change the reading frame for the whole rest of the protein. And this is why it can be more dangerous to lose or mutate 1 or 2 nucleotides than add or delete 3 nucleotides. It really can shift the reading frame. All right, let's test to make sure we know how to decode a messenger RNA. So here's a clicker question. Above are the first nucleotides of an RNA. Upon translation, which of the following peptides would be expressed? Ten more seconds. All right, final answers. All right, a lot of people like B. Who wants to explain their reasoning? Go for it.
So you go down to 5' and 3' until you find your start codon. So you go GC to G and then AUG. That AUG is the start codon, so we start with the lining. And right after that, it's proline.
Right, because it's C, C, U. That's their proline, and if you keep going, that's what the rest of it will be. Almost everyone put B, so I think we understand that first you need to scan until you find that first AUG. And there is no reading frame. Don't start reading in groups of 3. So one mistake that people sometimes make is they start in groups of 3, and if you do that, then this will be your first AUG. No, there is no reading of groups of 3. You scan along until you find that first AUG, and only then have you set the reading frame and you start reading in your groups of 3. Why was E incorrect? No peptide can be synthesized because there is a stop codon before the first start codon. Yes? Yeah, there can be no stop codon. There can be nothing until you first have the start codon. So if you saw maybe, oh, there's a UGA here, that's a stop codon. No, you have to first find the start codon, and only then do you start reading. Is that clear to everyone? All right. So let's talk a little bit about how these codons, these groups of threes in our messenger RNAs are actually read. And what they're being read by are the tRNAs. And the part of the tRNA that is going to base pair to the codon is the anticodon. And as always, it's always going to be that anti-parallel base pairing. So it's very critical for the tRNA to be attached to the correct amino acid. And this is what we call tRNA activation. That is simply charging of the tRNA or attaching that amino acid to the tRNA. This is done by specific enzymes called Aminoacyl-tRNA Synthetases. There are 20 different Aminoacyl-tRNA synthetases because there are 20 different amino acids. And so in this particular example for phenylalanine, there will be a pocket where only the amino acid phenylalanine fits. And then another pocket where all of the tRNAs that can specify phenylalanine will fit into that pocket. They will then be linked together, so the 3' end of that tRNA forms a high-energy ester bond with the amino acid, so the carboxyl terminal of the amino acid here. They get linked together. When that linkage occurs, there is a proofreading step where the synthetase can kind of feel if maybe the wrong amino acid happened to get in that pocket or the tRNA just structurally doesn't seem right. If that happens, that bond can be severed, and they can try again. But once it has been attached and released from the aminoacyl synthetase, this is the amino acid that is then attached to that tRNA. So the anticodon better be used for decoding the amino acid to which it was attached. So you'll have your tRNA, it has its anticodon, and as we'll see, when this enters the ribosome, it will be able to base pair to the codon site in the messenger RNA, and it brings along the amino acid to which it was attached. And this is how the correct amino acid will be selected during decoding. All right, so there is a property of codon-anticodon interactions that allows for what we call wobble base pairing. And wobble base pairing simply means that it is non-standard base pairing that is permitted, but only at the wobble position. For tRNAs, the wobble position will be the first nucleotide from the 5' end that is going to be base paired to the 3 position of the mRNA codon. So what does that mean? You use this kind of chart here to determine if in the 1 position I have a U, for example, then in my mRNA codon at the 3 position, I could have an A, that's the standard base pairing that we'd expect, but also G is allowed at that position. So a tRNA is able to base pair to either one of those positions. And so if I am asking you for the wobble pairing that is allowed, again, I'll always provide this kind of chart for you. You don't have to memorize which nucleotides base pair during wobble position. The thing that you do have to remember is which position in the tRNA versus the mRNA is allowed to wobble. So for the tRNA, it's going to be the first position from the 5' end, which means then it's going to be the 3rd position in the codon of the messenger RNA. Question? Pseudouridine can sometimes be in the anticodon, but don't worry about that for wobble right here. There is a nucleotide that we haven't talked about yet here. So this I stands for inosine. This is a modified version of adenosine. And in fact, if that is in the wobble position of the tRNA, you can pretty much base pair to A, U, or C. So is this clear to everyone? Let's do a clicker question and make sure it is. So if the anticodon of a tRNA is 5' GAU, what are all of the possible mRNA codons that can be recognized?
Oh, yes.
All right, looks like most of you have clicked in already. 5 more seconds. Another strong bias for letter C. Who wants to explain how they figured this out? Yes. Absolutely, yes. So the answer is indeed C. You have standard base pairing at these positions. Those are not your wobble positions. Don't do anything there. Do your normal base pairing. So U is going to always base pair to A is always going to base pair to U. But then we recognize that this 5', this first position from the 5' end in our tRNA, this G can base pair to a C or could base pair to a U. Everyone seems to understand that. I think we're mostly correct. And then also you could look at it the reverse way. So I could also give you a table and told you that if this was the codon of the mRNA, then the anticodon could be all of these different pairing nucleotides at the one position in the anticodon. Again, I would give you this kind of table. The thing you have to know is it is going to be the first nucleotide from the 5' end in the anticodon of the tRNA, and therefore the third position of the codon from the messenger RNA. Any questions about that? All right, so now we're ready to move on to actually talk about the stepwise synthesis of proteins by the ribosome. And so first we will look at these ribosomal subunits, what they consist of, and then talk about the 3 specific steps that translation undergoes to make protein. All right, so eukaryotic ribosomes come initially in 2 separate subunits. There is what is called the large subunit, consists of about 50 proteins, and the 3 different ribosomal RNAs known as 28S, 5.8S, and then remember 5S is that one that was synthesized by RNA Polymerase III, but it is also part of this large ribosomal subunit. And then the small ribosomal subunit is smaller, it has less proteins, it only has 1 ribosomal RNA, the 18S. Again, these S's are sedimentation values. 60 and 40 does not add up to 100 because it's based on how it's going to sediment. Here is a structural picture of what the translating ribosome actually looks like. Oh, wait, why is it 50 and 30? I thought it was 60 and 40. What's the difference here? Exactly, yes. So what I showed you in the last slide was the eukaryotic Prokaryotic has a little fewer proteins, and so the sedimentation values are a little smaller. But prokaryotic ribosome translation essentially works the same way eukaryotic translation does. And this is why we can translate human proteins in bacteria cells. What I want you to appreciate from this structural view is see all this coiled part, this white coil, gray coil down here. That is all the ribosomal RNA. And so you can really appreciate how a really large fraction of the ribosome is this ribosomal RNA, these long ribosomal RNAs. All right, so how do we initiate translation? So to initiate translation, we need a special tRNA called tRNA, which stands for initiator tRNA. We'll see why we need this initiator tRNA. It encodes for methionine because we're going to look for that first AUG, which encodes for methionine. The reason that this is a different tRNA than the one that's used if we're putting methionine in the middle in a RNA that's being translated is because it's the only tRNA that fits into what we call the P site of the ribosome. And we'll talk a little bit more in a couple of slides what these E, P, and A sites are. But for now, all you need to know is that for translation initiation to start, we need to have the initiator tRNA attached to methionine amino acid. There are initiation factors, or IFs. So this 2 is IF2, for example, that binds to the tRNA, as well as the small ribosomal subunit. And so these all come together in a complex to then find the 5' end of the messenger RNA to start looking for that start codon. All right, so remember when we said the 3' end that has that poly A tail of a messenger RNA gets bound by poly A binding proteins and that this is important for translation? One of the reasons is this poly A binding protein actually helps stabilize the binding of initiation factors to the 5' cap. So that structure at the 5' cap is recognized by translation initiation factors, and it's kind of stabilized there by interactions with poly A binding protein. The mRNA, at least in some cases, sort of loops around to form this structure. These initiation factors, and you don't need to memorize the various numbers like EIF4E, 4G, 4A, 4B. Just remember IFs for initiation factors when we're talking about translation. So there is a group of initiation factors that recognize the 5' end of the messenger RNA with some help from the poly A binding protein. This then helps recruit this complex that contains the small ribosomal subunit, the initiator tRNA methionine that is attached to methionine, and some more initiation factors. This complex, once it then joins the messenger RNA, is going to start scanning along that 5' UTR. And remember, this tRNA has an anticodon. It's looking to base pair to AUG. And so it's going to scan along until it finds that first AUG. Once it base pairs, the anticodon base pairs to that AUG, the whole complex is going to pause at this start codon. That pausing then allows the other subunit. Remember, we still need the other half of the ribosome to come in. So this large 6DS ribosomal subunit is then going to join the complex. There, they get locked together to then form the ADS. And this pausing step, as well as this locking together, these are accompanied by hydrolysis of GTP through the action of some initiation factors that are part of this. Once ADS has formed, we now have ended the initiation phase. We now have a ribosome that is ready to start elongating for translation to begin. So the next step is going to be translation elongation. Before we talk about that, let's talk about these 3 different sites that are in the ribosome. So This P site, which is the where the initiator tRNA can initially bind to, that is called the peptide site, and that is because that is where the growing peptide, the chain of amino acids hooked up to the tRNA, is going to be found. The E site is called the exit site, and you'll see why. That's where the tRNAs, after they have given up their amino acid, are going to be shifted to the exit site so that they can be kicked off the ribosome. Excuse me. And then the A site is called the aminoacylated site. This is the site where the new incoming tRNA that is attached to the amino acid, it is aminoacylated, so amino acid number 2 shown here, will come in, and this tRNA's anticodon will sample whether it can then base pair to the codon that is in the A site. So that is what we mean about these 3 different sites. All right, so we already have our initiator tRNA in the P site. We have the empty A site, so the tRNA can come in, try to base pair its anticodon to the codon. If that base pairing is accommodated and the tRNA fits correctly, there's going to be GTP hydrolysis. Just how like we had initiation factor, those IF proteins that were helping with initiation, during elongation we have EF proteins. And again, don't worry about the numbers and the alphas and all that. Just know that they're elongation factors that help bring the tRNA in and lock it into position. We now have the tRNA that is attached to its amino acid in the A site. We have the tRNA initiator that is attached to methionine at the P site. And so now we're ready to do the catalysis to actually form a peptide bond between those 2 close amino acids. And so the enzyme activity is a peptidyl transferase reaction, and we now know that that is indeed catalyzed by the ribosomal RNA. It is a ribozyme. It is the RNA that is catalyzing the peptide bond formation between these 2 amino acids. When that peptide bond formation occurs, the second amino acid stays attached, its C terminus is still attached to the tRNA, but now amino acid 1, that high energy bond has been severed because it is now in a peptide bond with the amino acid attached to the tRNA that is in the A site. All right, well, what do we have? We have a growing peptide bond here. That should not be in the A site anymore. And so there needs, this is just to point out that this is the catalytic core, and that's where there's this ribosomal RNA that's able to do this peptidyl transferase reaction. All right, so we have the growing peptide bond on the tRNA that is in the A site, so we need to move it to the P site because that's where it stands for peptide bonds. And so there is a translocation event where the RNA moves so that the growing peptide bond on the tRNA is now in the P site. This tRNA that gave up its amino acid, so it's empty, it's no longer aminoacylated, that gets shifted into the E site or the exit site. And now we again have an empty A site. Next step that's going to happen, next tRNA is going to come in, try to base pair its anticodon to the codon, and the process is going to keep going until a stop codon is reached. Once there is a stop codon in that A site, there are no tRNAs that read the stop codon. Instead, we have what are called release factors. Release factors are proteins. They are made out of amino acids. And there is one called release factor 1 that is actually shaped like a tRNA. Why is it shaped like a tRNA? Well, it needs to fit into this A site, which normally all the tRNAs are coming into. So this RF1 shaped like a tRNA, but it recognizes when there's a stop codon on there. It's not base pairing. We're talking about a protein here, but it comes in, recognizes there is a stop codon sequence here. It recruits another release factor called RF3. RF3 brings in GTP, causes GTP hydrolysis. That hydrolysis then allows for several things to occur. One of them is the association of a protein called ABCE. This is important because it causes the 40 and 60 subunits to break apart. This then allows release of the messenger RNA as well as release of that new peptide. That new protein can then complete folding and go off. And what do we have now? Well, we have the RNA that can undergo multiple rounds of translation. It can be decoded again. We have the two freed subunits. So now we have this small ribosomal subunit that can go back, start initiation, and do the whole process over again. Any questions about these 3 steps of translation? Yes?
So are tRNAs that just constantly go into the base and checking, or are initiation factors recognized?
Yeah, so the recognition really is by the base pairing. There are some ideas that the codon even provides a little bit of information to help the shape of the A site accommodate certain tRNAs better for that. It's not entirely clear how that works, but yeah, there's just so many tRNAs that are aminoacylated and they are sampling. And it's very, very rapid. But it also needs to be really accurate too. And so most of the time they get it right. The last thing I want to mention is 2 features of translation that are thought to make it more efficient. The first is that you can have multiple ribosomes translating the same messenger RNA. We call this a polyribosome, or sometimes polysome for short. What that means is as soon as you start initiation here with your small subunit and the initiation factors and start scanning for that start codon, and then you recruit the 60S to make the full 80S and start translating, as soon as that moves away from the start codon, a next one can come in and a next one. So one messenger RNA can have multiple translating ribosomes on it. The ones that are furthest from the start codon are, of course, going to have the most amino acids. They have translated the most amount of messenger RNA. And so this is what we call a polysome or polyribosome. We also think that by the poly A binding protein interacting with the 5' end of the messenger RNA with these initiation factors, that aids in initiation, but it also potentially brings the stop codon closer to the 5' end of the messenger RNA than if it were in its long linear shape. And what that means is when the ribosome splits, it's potentially closer to the 5' end to allow for more efficient reinitiation by these translation factors. Any questions about that? Yes? So there are a lot of features, especially ribosome or especially RNA binding proteins, that can regulate how efficiently an RNA can be translated. Some of them will have less multiple ribosomes on them because they are weakly recognized for translation initiation. Others, where you need to make a lot of protein, are much more efficiently translated, and you might have a lot of ribosomes. If you have a messenger RNA that's relatively short, has a small open reading frame, you might only fit 1 or 2 ribosomes on it at one time. So it varies for different messenger RNAs, yes. All right, so translation is essential, right? We're making proteins all the time, and so it's sometimes surprising to think that can there be disease states associated with these translation factors, and what's going to happen if translation efficiency is altered? Turns out that there are a lot of diseases that have actually been mapped to mutations in various factors that are involved in translation. And again, this is not something for you to memorize, but just to appreciate that a lot of what we've learned about translation has come from studying genes that, when mutated, change the efficiency of translation. And of course, this can lead to various disease states because it's important to have good translation going on. Turns out also that a lot of cancers change either the levels or the activity of translation factors. In many cases, it increases their efficiency. Why would a cancer cell want to have more translation, more rapid translation, more protein production? Yeah, cancer cells are just rapidly dividing, rapidly growing. If you're growing a lot, you need a lot of proteins. And so in many cases, cancer proteins have found ways to make translation efficiency work harder. And the main point, of course, about understanding translation and the factors that are involved in it is then we can start to come up with therapies. If we know what's going wrong, if we know which factor is overexpressed or overactive in the cancer cell, we can start designing various therapies to target that. And again, this is just to appreciate, how far we've come here. And if you're interested, there's a review. You can read more about this. And then the last thing I want to mention as to why it's so important to learn about the mechanisms of translation is this turned out to be very useful when we were fighting COVID, right? So the first vaccines that probably all of us got were these RNA vaccines. The reason we were able to do this is because we understood how our cells translate messenger RNA to make proteins. We designed an RNA that encoded a viral protein, got it into our own cells, and our own cells, our own ribosomes, took that RNA, made, for example, the spike protein for COVID. That spike protein was recognized by your body as, ooh, this is a weird protein I've never seen before. I better make some antibodies for it. And therefore, when you get infected by the real virus that's all coated with spike proteins, the antibody stores you already had said, aha, this is foreign, and I can help get rid of this virus faster. And that was all based on understanding how to make an mRNA that your cells will translate robustly into a protein. And that is it for today.